Live. So good morning. This is a quick sprint to the finish of the high-level material, and we're going to do respiration, the last stage of respiration, electron transport chain, and then we're going to go through photosynthesis. Here we go. So we've talked about the first three stages of respiration, and now we're into the last one, electron transport chain. Now, if you remember, we haven't really earned a lot of energy along the way. Very little ATP has been produced. This is the payoff stage. This is where you get all the ATP. What you do have is reduced NAD and FAD, which are electron carriers. So you've got all these electrons, remembering they're not just electrons, it's an electron and a proton, a hydrogen ion, which is useful because we will need those hydrogen ions, as we will see. So the last stage is where most of the ATP is being made. So 34 ATP, you may find different values on different websites because in reality there is some debate over exactly how much ATP is made. So making ATP from the electrons is called oxidative phosphorylation. So this is a name because basically you're using oxidation to turn ADP into ATP or phosphorylating it. So overall this process could be called oxidative phosphorylation. So I've just said that ATP, you're adding phosphate. Okay, two things are going on. So first of all, the energy in the electrons is being harvested, the electron transport chain. Okay, this is like you're getting the energy from the electrons, and those are the electrons in the NAD and FAD have brought. And secondly, the energy is used to make ATP. Now the way this works is you pump so many protons into a tiny space that they flow out through the specialized protein channel that makes ATP. This tiny space, by the way, is the space where the inner membrane of the mitochondria. If you remember that there's an inner membrane and an outer membrane, it's a double membrane, so you pump it into the intermembrane space and it flows back out again into the matrix. That is your little tiny space. So there's folds in that inner membrane. We'll look at that in the diagram. And so there's a high concentration of protons and they flow out by chemiosmosis from a high concentration to a low concentration. And it's kind of like water thrown through a turbine in a power plant. Okay? As they flow out, they make energy, and that energy is used to turn ADP to ATP. So,
is it associated with the phospholipid?
Just speak up so they can hear you. Go on.
Is it associated with the phospholipid bilayer?
It is a phospholipid bilayer, but it's not the phospholipid bilayer that drives chemiosmosis. It's not. It's actually a protein channel. So picture your phospholipid bilayer. It is there. There is a special protein channel. And the hydrogen ions go through the protein channel, not through the phospholipid bilayer. And that protein channel is modified so that as the hydrogen ions flow through it, they cause ATP to become ATP. So, and it's kind of an indirect process, as we'll see. The energy from the electrons is used to pump the hydrogen ions in there. That's active. The hydrogen ions flow out, which is passive, and that causes ATP to imagine ATP. So imagine like we had water in a dam, and I pumped all the water from a river into a dam. That's kind of like harvesting the energy from the electron transport chain, using it to pump the water. And the water's going to flow back out by gravity. When it flows out, it goes through a turbine and makes your energy. It's kind of like that metaphor. You pump the hydrogen ions in using the energy and they flow out passively, but that generates energy. So, in the electron transport chain, a series of electron carriers, these ones don't move, like NAD and FAD. So we've got other electron carriers here. But they're not mobile ones. They're embedded in the membrane, the inner membrane of the mitochondria. That pass electrons between them, releasing energy for chemiosmosis. Energy is released because the electrons go to a lower energy state as they pass from one carrier to the next. Imagine a line of people passing a hot potato. Okay? And the potato gets cooler as it goes from one person to the next. A long line of people, first person, the potato's super hot. Okay? They throw it to the next person. Bit cooler. Why? Because heat is passed from the potato into their fingers each time. So it's a little bit like that. Okay, these electrons are going from a high energy state, like they're hot, and then as they go along the carriers, it gets lower and lower and lower. The electrons are finally accepted by oxygen with hydrogen to make water. This is important because we remember that when you breathe in, that oxygen is used to make water. The oxygen you breathe in. There's oxygen in glucose. But that oxygen is used to make carbon dioxide. We've talked about this before. Okay, this So the final electron acceptor, like the final person carrying the hot potato, is oxygen. Okay? And that also combines with protons, H+, because you've got plenty of protons that have been flowing out for the protein channel to make to cause ADP to turn ATP to make water. So that's why you make water here. And I think this is a point which often comes up in questions, in exam questions about respiration. What's the role of oxygen? Oxygen is the final electron acceptor in the electron transport chain. Okay, so this is where the water comes from as a product of respiration. Right. So, chemiosmosis, I'm just going to scroll down a bit here. So chemiosmosis. So it's kind of like osmosis. It works like osmosis, but it's not water. Okay? It's chemicals. It's a buildup of electrons and subsequent release through a protein channel from high, sorry there's a typo there, it's high not height, high to low concentration to generate ATP. So, what we're doing is we're pumping them into the intermembrane space. So that's the space between the inner and the outer membrane. I'm just going to scroll down for a second here. Take a look at the mitochondria. So if you have a look at the mitochondria, you can see there's an outer membrane, there's an inner membrane. Okay, this is where all the action happens here. This is where the protein channels are. There's a space between them. This is the intermembrane space. They're being pumped into this space in chemiosmosis, and then they'll flow out again into the matrix. Just taking a closer look. So this happens by pumping into the intermembrane space using the energy released by the electrons in the electron transport chain. So where does that energy come from? Okay, the energy comes from those electrons, the hot potato analogy I've used. As it gets cooler, imagine that every person could use a bit of that hot potato heat to pump these electrons across the space. That's what happens here. So, using energy from electrons, hydrogen ions in the matrix go to hydrogen ions in the intermembrane space. I like to show you this. So the hydrogen ions are here, and they're being pumped up into this intermembrane space using the energy in the electron transport chain. The electron transport chain is lined up along here. Okay, the electron carriers which pass the electrons, the hot potato analogy, it's happening on this in the membrane. I've got a photo of what it looks like here. Two channels for hydrogen ions open in the cristae, and highly concentrated protons are allowed to flow out. As they do so, they drive the production of ATP from ADP to P. So let's take a look at a picture here. This is the inner membrane, mitochondria. So you've got, first of all, you've got channels which you can pump hydrogen ions in. So hydrogen ions are pumped in, and you've got NADH and NAD, they're bringing the electrons. The electrons are going to be passed along between the protons, and they're going to be high energy electrons. And the energy they have is being used to pump hydrogen ions into the membrane space. They're the mobile electron carriers. Okay, now actually we're using the word electron carriers again here, because these are the electron carriers that from the previous stages, the Krebs cycle, okay, glycolysis, they've brought electrons and protons, and they're oxidized, they release them. And these are the electrons here, which are actually being passed along.
Oh, are those proteins like active transport that requires electrons?
Yes, they're the two things. Basically, this is you're quite right. This is active transport. It requires energy, and the energy comes from the energy electrons. The electrons are passed between them. The energy is harvested and used to generate energy to pump hydrogen ions across.
And from that you gain
And you haven't got ATP yet. What you have got now is you've got huge concentration of hydrogen ions in the intermembrane space, and it's a low concentration outside. So naturally, by diffusion, they flow out. Just as if you had a lot of water in a dam,
it just flows out.
And it would just flow out. Okay, because the water level would be higher inside the dam than outside the dam.
That's why it's called chemiosmosis.
Yes.
It works like osmosis.
It kind of works a bit like osmosis.
But it's with chem.
Yeah. And this is a specially modified protein channel, okay, that allows hydrogen ions to flow through. and as they flow through, they turn ADP to ATP. And what's driving this is actually the chemiosmosis itself, the movement of the hydrogen ions from a high to low. Now note this is passive. This is active. It's kind of like you've invested your energy here and then you get it back, you get your ATP. It's kind of like if you imagine you pumped water into a dam and then the water flowed out, and you don't really make the hydroelectric power until it flows out.
How does ADP becomes ATP?
How does ADP become ATP? So you've got ADP is adenine diphosphate. It has two phosphates, and making that bond, what you do is you add another phosphate. You can see there's a free phosphate here. And that bond between two phosphates is very energy rich. It's energy rich and it stores energy. Breaking that bond releases energy.
But does it need hydrogen proton in order to make that bond?
It doesn't, but there's some, it's actually, you're asking a question which is really complicated. It's to do with the structure.
Okay.
So it's to do with the actual structure of this protein molecule. I've looked at diagrams of it, but as the hydrogen ions come through, it causes the ATP to...
Do I have to know to that
You
level?
don't need to know to that level. That's cutting edge, cutting edge. That's, yeah.
Okay.
This is enough. It's
So it's
enough.
like by pulling in
Yeah.
hydrogen again from the highly concentrated inside the inner membrane, ADP becomes ATP.
Yeah,
I just have to know that.
yeah.
Okay.
It actually, it turns. It's really weird. I'm like, I've looked at diagrams
The
of it,
diagram
but I
is
was like,
really
wow,
complicated.
it's complicated. As far as we're concerned at this level, I would like you to think of it a bit like the flow of water. So the hydrogen ions goes from the intermembrane space
So,
back into the matrix,
okay.
high to low concentration, and it drives ATP being turned to ADP.
Do I have to know like the
And-
specific proteins, 1, 2, 3, 4,
And
the roles of that?
sorry,
The roles of the specific proteins, the active proteins.
are these?
Yeah, because it has the numbers.
No.
I don't accept
No,
it.
you just think of them as being an electron transport chain. In photosynthesis, we need to do a little bit more. For this point, they're electron carriers. They're in a row. Okay, you call it the electron transport chain. And look, there's the electrons that go there, and then there. And then you've got hydrogen, which has been flowing out. So you've got hydrogen, electrons, put them together with oxygen, and you get water.
Water. That's why the oxygen is the...
Yeah. I just want to show you this video, just to summarize what we've been saying. This is funny. Well,
Welcome to the story about cell energy. The goal is explaining how cells make ATP. It happens in the mitochondria, which you can think of as the cell's energy factory. Mitochondria are double membrane organelles, an inner membrane and an outer one as well. The mitochondrial matrix is the fluid inside. It's where reactions like Krebs cycle reside. Glycolysis and Krebs make NADH and FADH2 from energy and food.
I'm not sure about the subtitles though.
Oh, you can click CCL.
Yeah, let's get rid of those.
These electron carriers make their way from the matrix to the inner membrane. And that's where you find the electron transport chain. It's a series of enzymes embedded in the membrane, which take the electron carriers' electrons and uses their energy for pumping protons. The mitochondrial electron transport chain uses electron energy for pumping protons from the mitochondrial matrix to the intermembrane space, increasing proton concentration in that place. The only way the protons can escape is through a channel and an enzyme ATP synthase, which uses the fused protons' kinetic energy to make ATP from ATP and P. The chain is a series of enzymes in a row. Each accepts electrons and lets them go to the next carrier in this transport chain. It's kind of organized like a bucket brigade. What drives electrons down this enzymatic series is the growing level of each carrier's electronegativity into oxygen. Electrons ultimately fall. It's the most electronegative of them all. It's NADH that starts this run, donating electrons to complex number 1. This powers active transport as protons are displaced and get pumped from the matrix to the intermembrane space. From complex 1, electrons flow to ubiquinone, also known as Q, which floats through the inner membrane happily and brings its electrons to complex 3. And you can guess the function of complex 3. It's another proton pump using electron energy. Protons jam up in that intermembrane compartment like 100 people in a one-bedroom apartment. The mitochondrial electron transport chain uses electron energy for pumping protons from the mitochondrial matrix to the intermembrane space, increasing proton concentration in that place. The only way the protons can escape is through a channel and an enzyme ATP synthase, which uses diffused protons kinetic energy to make ATP from ADP and P. Complex 2 is for FADH2, which donates electrons, which then get passed to Q, which once again passes them to complex 3, which pumps protons using electron energy. From complex 3, the electrons proceed to another mobile carrier cytochrome C, which donates the electrons to complex 4. Another proton pump, could you ask for more? After complex 4, electrons flow to oxygen, which is ever so electronegative that pulls electrons down the chain, keeping them moving like the cars of a train. And NaSO2 does this electron grabbing trick. It also grabs protons from the matrix. They all combine to form H2O. Electron transport chain, watch it go. The mitochondrial electron transport chain uses electron energy for pumping protons from the mitochondrial matrix to the intermembrane space, increasing proton concentration in that place. The only way the protons can escape is through a channel and an enzyme ATP synthase, which uses diffused protons kinetic energy to make ATP from ADP and P. Now all these protons in the intermembrane space are trapped. They can't get out of that place, cause protons are charged and could never get through a phospholipid bilayer. They can't diffuse, but like all particles, they're dying to go from where the concentration's high to where it's low. Stuck in the intermembrane space, they're frustrated to diffuse to the matrix. They're highly motivated, and this gradient's been made steeper by O2, which absorbs protons from the matrix stew. So from proton pumping and oxygen's actions, add another force, electrochemical attraction. Think of all those trapped protons, each one's positive. The matrix in comparison is negative. Opposites attract, so the protons are dying to get to the matrix. Oh, how they're trying. There's only one channel that lets protons pass, and they use it like high school students busting out of class. It's a channel and an enzyme. It's ATP synthase, the closer in this game, an energy ace. ATP synthase is embedded in the inner membrane. How it works is so cool, it's insane. It's got channels for diffusing protons running right through it. When cells make ATP, well, watch how they do it. The matrix side of ATP synthase has binding sites for ADP and P, which come in and bind. And as ATP synthase lets protons barge through, their kinetic energy gets put to use. Like water through a turbine, proton movement generates rotation, change in synthases, binding site conformation, which catalyzes chemical bond formation. ADP and P make ATP that energy sensation. The mitochondrial electron transport chain uses electron energy for pumping protons from the mitochondrial matrix to the intermembrane space, increasing proton concentration in that place. The only way the protons can escape is through a channel and an enzyme ATP synthase, which uses diffused protons kinetic energy to make ATP from ADP and P. The mitochondrial electron transport chain uses
It's unusual, but it's actually brilliant, isn't it?
Yeah, it is.
It's
That
really nice.
structure, like, if the passive thing, that's like crazy. That making ADP to ATP.
That
is
is,
actually
it
crazy.
is really, it's crazy. But note, there's one thing that I didn't mention earlier that the picture in the video was that proton channel is an enzyme. It's ATP synthase. But they're all enzymes, and it mentions some things like quinone, like complex 1, complex 3, complex 2. They won't ask you to recall that, but if they throw it into a question, you've seen it once before, you're aware of it.
You can throw it with the essay question.
Yeah, I think it's nice, that video to look at.
Just call that Q thing. With
Yeah,
just
quinone,
letter
Q, they use it,
Instead
yeah.
of come C with side
Yeah,
C.
we can use the abbreviations. You can say ATP instead of adenosine triphosphate.
Without saying
You can say that. You can say ADP.
Okay.
Okay, just to look. This is a nice little summary page we had earlier of what's happened, everything we learned of respiration, and the last little thing is just to look at the adaptations of mitochondria. Mitochondria adapt to respiration the following ways: there's a double membrane that helps the mitochondria control the environment of the matrix and ensure conditions are optimal for the Krebs cycle. The inner membrane is folded to form the cristae. Okay, those are the little folds. This is where the action happens of the electron transport chain and the intermembrane space. Okay. Because it's extra small, we're looking at the intermembrane space. It's not so much here, but like here, that folded cristae, this folded particle. This is a really small space. That's where a lot of the action's happening. The instant membrane space is small. This makes it easy to build up high concentrations of hydrogen ions that drive chemiosmosis. These features have been understood by using tomography, which is like a three-dimensional picture by overlaying 2D images from a transmission electron microscope. So imagine if you took a 2D electron microscope image, like a scanning electron microscope image, and then you made many of them and overlaid them. You can build like a 3D image, and that's how we've understood this. That technique is called tomography. Remember, mitochondria have their own DNA and a ring on their own ribosomes. So this is suggestive that they're of the endosymbiotic theory, which is the idea that they were originally prokaryotic cells that became incorporated. Exam question here. Distinguish between oxidation, reduction, and biological reactions. State two products of glycolysis. Explain the role of cysti in mitochondria. So,
oxidation and reduction. Do I just distinguish?
What do you think?
Oxidation is like when you gain electron.
Actually, the opposite.
Oh, is it?
Yeah.
Okay.
So remember oil rig. Oxidation is loss.
Oh, oil rig.
Loss of electrons, reduction is gain of electrons.
Yeah.
Oxygen is, you gain oxygen. You react with oxygen. Oxidation, react with oxygen. Reduction, oxygen is often taken away. Hydrogen is the opposite. Lost or gained. Energy loss. I would remember oil rig. Okay, oxygen, oxidation is loss. Reduction is gain of electrons. It's a nice way. Okay, two products of glycolysis.
It created ATP, 2 ATP.
Yep.
And NADH.
Okay, they'll accept that. ATP, NADH. Also, you get pyruvate. You get the 3 half. So that's nice. You would have got that one. This one here. Explain the role of cysti in mitochondria.
Cysti.
So cysti are these folds here.
Oh,
The
it's
folds in the inner membrane are called cysti.
So it's where the chemiosmosis happens.
Okay, we got that. Anything else?
Is it like the acid question?
Yeah.
Okay, then I have to explain like with the complex channels, like
Yeah,
the one that I learned.
you could talk about the transport chain. So
the
let's.
NADH and FADH
Okay.
donates the electron, and then the protons gets to the inner membrane space, and then it gets pumped out by the highly concentrated ions with the passive, by passive way with ATP synthase.
So if you get asked about this, you can throw in sort of the angle of the surface area, because if you have a high surface area of the inner mitochondrial membrane, that means you've got more space for everything. You've got more space for electron transport chain. Okay, allow electron transport embedded. But this is what you said. Embedded protein carriers allows electron transport chain. That's what you just said. Facilitates protein pumping because of high surface area, again, and then increased ATP production because ATP synthase embedded in the membrane.
Basically like how it is doing.
Yeah, it's the fact that you've got that extra surface area to do that. Okay, brilliant. That's so that's the end of respiration. Let's look at photosynthesis. Whoops. Okay, photosynthesis idea. So the idea is basically you want to understand the difference between the light-dependent and the light-independent reaction. Detail the steps of the light-dependent reaction, detail the steps of the light-independent reaction, understand how chloroplasts are adapted to their function. So quite a similar idea here. Right.